All right, guys, today let's talk about the cell cycle. All right, but before we do that, let's go ahead and let's review over the DNA stuff that we've talked about so far, okay? So DNA, let's talk about what it stands for. DNA stands for deoxyribose nucleic acid. Okay, that's what it stands for. And then my function of DNA is, don't forget, to store genetic info. And what is that genetic info coding for? Remember, genetic info is going to code for some type of protein, say some type of part of your physical organism. His monomer is a nucleotide. And remember, nucleotides are made up of 3 parts. You have that phosphate, so that backbone that's really, really sturdy, I suppose. And then you have a sugar in the middle because he's sticky. That's where the deoxyribose comes in. And then you have your 4 nitrogen bases. Also, DNA is a double helix, which means it has 2 strands. And what we kind of want to look at today, we're going to talk a little bit about the double helix part again, but we also want to talk about how DNA, when I want to make a copy of DNA, remember usually I want to make a copy because I need to make new cells. When I want to make a copy of DNA, that's done during replication, and that's when I use Chargaff's rules, like the A's and the T's go together and the C's and the G's go together. Okay, but DNA is kind of like if I said H2O, right? H2O, how can I see H2O? Well, easy enough, you're going to say water, but what do you mean by water? You mean water is liquid, right? You can see water as a gas, and you can see water as a solid, right? And so you still have this H2O. You guys know what H2O is, but I can see it in different forms. Well, it's the same thing with DNA. DNA is just a nucleic acid, but guess what? I can see it in different forms, okay? And so we're going to go through those forms. The first form is generally like when your DNA is just sitting inside the cell, right? DNA is generally in a loose state. Like if we drew a cell, right? It doesn't matter what kind of cell it is, but here's my cell. When we drew DNA in the middle, normally we would just kind of do something like that. It's in a loose state. Why it's in a loose state? Oh dear. Why it's in a loose state, this makes it really easy to copy. You know, it's kind of a far-fetched thing, but if you were to take a book and copy a page out of it, if the pages are really loose, then you can fan through them, and then you can find like maybe that particular part that you need to copy, right? So it makes it really, really easy. When it's in this loose state, this is called chromatin, okay? Chromatin is loose state DNA, all right? I kind of like to look at it, you know, if you're a visual person, I think this looks like a plate of spaghetti, right? And so here's your plate right here, and then here's your spaghetti in the middle. But here's the deal, when you need to eat your spaghetti, you can't eat it when it's loose like that. Like what do? You always take your fork and then you spin it around, right? So whenever we need to move my DNA, moving DNA like that, that's a little bit harder, right? And so when you need to move this DNA, that means it's going to coil up, right? So if this is your spaghetti and you want to eat your spaghetti, then you twirl it on your fork so you can bring it up to your mouth. That's what this is doing. So when you move your DNA, you're going to coil it up. But if you think about it, when you coil up your spaghetti, it doesn't just spin on its own. You have to have a fork. And so for my DNA to coil up, that means it's going to wrap around these histone proteins. Okay, that is the thing that's going to allow it to coil up in the first place. All right, what that looks like is, we can go and use that green. So what that's going to look like is, instead of it being loose, now it's in this nice little tight package, right? And these red things, those can be those histone proteins, okay? So it's wrapped around there. So when you guys eat your spaghetti, you have to wrap it around a fork. That fork is your histone protein, all right? This has a name. This is called a chromosome, okay? This is called a chromosome. So chromatin is when it's loose, and then a chromosome is when it's coiled up, right? But don't forget, you know, if I'm doing all of this, I'm probably making a new cell since I'm moving it, and that means I've replicated it, right? So since the DNA has been replicated, I don't have one chromosome. How many do I have? I have 2 chromosomes. Okay, I have 2 of these suckers. If I look at it, so instead of it being 1 stick, now I have 2 sticks because I made a copy of it. And here's my histone proteins there in the middle, right? So what's this guy's name? Well, if I've replicated my chromosome, this is now called a, whoops, switch my eraser. This is now called a replicated chromosome, because that just means it's been doubled. But normally when you see it like this, you don't really see it drawn like this. Generally, this is drawn as an X, okay? So this is now my replicated chromosome, is the X, all right? Now there's a couple different structures on this replicated chromosome that we need to look at, all right? So let's talk about my replicated chromosome. Okay, replicated chromosome. Again, he looks like an X. But if we go back to the spaghetti analogy, have you ever seen those really fancy restaurants when they use a spoon underneath the fork to twirl it? Well, they use that spoon so that way the spaghetti doesn't fall off. Well, this DNA, remember, it's just big and stringy, and I've coiled it up on some proteins. I don't want it to unravel if I'm moving it around. And so it has these caps on the top, okay? These are caps. These are called telomere, okay? Telomere. And a telomere is a lot like, oops, if I can spell it right. This is a lot like an aglet. You guys know the aglets on the end of your shoelaces, so that way it doesn't come unraveled? That's what the telomere does. So the telomere, this is going to keep the DNA from unraveling, right? And the reason I have it coiled up in the first place is because I'm moving. Now, I also want to keep this sucker together. I want to keep that replicated chromosome close. And so here in the middle, I have this nice little button, okay? This nice little button in the center is called a centromere, right? So a centromere, this is in the middle, okay? So centromere, it's in the middle, and it keeps my replicated chromosome, together, especially when I'm trying to move this sucker. All right, so DNA, a couple different forms. I have chromatin, which is loose. I have a chromosome when it coils up, and then a replicated chromosome, because normally I'm doing all this when I'm trying to make a copy of a cell, which is where we're going next. So let's talk about the cell cycle. And if we think about cell cycle is a lot like a life cycle, right? It's just the thing that your cells are going to go through. And so my cell cycle, what we have to think about is organisms need new cells, okay? And let's think about it. Like, why would I need new cells? Like, why? Like, why would your body make a new cell, right? So let's think. Why are you going to make a new cell? Well, you guys were a baby at some point, and now you're bigger. What did you do? You grew, right? And so sometimes I need to make new cells when I want to grow my organism. We've all fallen off of a bike before, or we scraped our knee or something. So we do have to replace cells at some point during our lifespan. And we also have to repair cells at some point. And so that's why my cells will go through a cell cycle, right? So when I go through a cell cycle, again, like a life cycle, when I go through a cell cycle, my entire goal, my function is to make new cells, okay? Like, that is my entire goal of the cell cycle, is to make new cells. So now we get to go through the stages of this sucker. Let's make this nice color. All right, so cell cycle. This is going to have 3 stages. It has 3 stages right there. All right, the first stage, this is called interphase. And I always like, because, enter, it's not spelled right, but if I enter something, that means I go into it, I start, right? So when I enter the building, I start school. So my interphase, this is my starting phase. And then phase 2, this is my This is my M phase, and M is going to stand for a couple different things, and we'll talk about that. And then 3, my last stage, this is cytokinesis, and we'll talk about what that word means too, okay? So let's talk about interphase first. Let's say this nice lilac color. Interphase. So I'm entering my cell cycle. It's the very, very first stage. My entire goal of interphase, my entire function, is to grow, and I want to double my cell contents. Which makes sense, because if I drew a silly little graphic over here, like here's my cell, here's a DNA, and then here's an organelle, right? And if I'm going to split this cell into 2, if I left it as it is, I would have half the DNA over here, half the DNA over here, or maybe I had a DNA in one, right? Maybe we could do that instead. I'd have DNA in one cell, and maybe I'd have an organelle in another. Are these 2 cells going to function if I only have DNA or if I only have organelles? No, of course not. But, so if I erase those really quick, but up here, if I were to make a copy of my DNA and I were to make a copy of my organelles, then when my cell splits, look, I can have DNA and organelle in one and DNA organelle in the other. So that's why interphase, I want to double my cell contents. That way when my cell splits, I have the right amount. Now my interphase is divided up into 3 stages. I have G1, S, and G2. All right, G1. G doesn't mean this, but this is when my cell is going to grow, right? My cell's physically going to get larger. That way when I split, I have something, for a cell to be made of still. So my cell's going to grow during G1, and I'm also going to double the organelles. Remember, when we start talking about eukaryote cells, which is what this cell cycle is, we're talking about a eukaryote cell, like an animal or a plant cell or a fungi cell. We like division of labor, correct? If I divide up the job, then my job is easier. So if I double my organelles here, that means I'm just concentrating on organelles, and that's all my cell's going to do. Because then here at S phase, S stands for synthesis. And if I synthesize something, that means I'm making it. This is when I double my DNA. And remember, when I double my DNA, we've talked about it, that's just replication. That's when I use my A's, the T's, the C's, and the G's. Again, it's division of labor. I do double organelles during G1 so I can focus on that. And then during S, I double my DNA because, DNA is pretty important and I don't want to mess it up. So I'm going to put all my concentration on that. And then G2, well, G, that's going to be grow. And then also, as I'm growing, I'm going to get ready to divide. Right? Because if I'm doubling all of this stuff, then I'm have to divide eventually. But that's all interphase is doing. That's literally its job, is to double. It's to double my cell contents. But I have these separate stages so that way I can concentrate on one task at a time and I can do it well. Now, next one, my M phase. Now again, M doesn't stand for this, but I always like to think of M as movement. So I just doubled everything during interphase. Now I have to move it. So M, by definition, right, this is going to be the division of nuclear, again, we like to divide up our labor, of nuclear contents. All right, well, what do I find inside my nucleus? I find inside my nucleus DNA. So M phase is purely about dividing my DNA, right? Completely about dividing my DNA. Now, I do have two different types of M phase. It depends on cell type. Okay, so the phase I'm looking at depends on cell type. If I am looking at like a normal body cell, then M stands for mitosis. And don't worry, we're going to go through all of that later. But if I'm looking at a sex cell, like a cell inside my ovaries or testes, then M is going to stand for meiosis. But they both start with M. That's why we just put M as general. And it's still moving my DNA. But depending upon what kind of cell I'm looking at will determine the actual phase. Okay, so interphase, I double. And then an M, I divide up my nuclear contents, which means my last stage, I have to divide up everything else, which is cytokinesis, right? So cytokinesis, this is going to be my final division. This is where I divide up my cytoplasm, because remember cyto, that refers to cell, and then kinesis. Kinesis means movement. And so my cell is literally moving, right? This is where my cytoplasm, remember cytoplasm, this is all cell contents, divide. So I've already divided my DNA. That's what I did during M phase. Now I get to divide everything else. And this is where I get two new cells. Okay? So let's draw this. I think that'll help a lot. So we have the words, now we just have to put the pictures to it. Let's go ahead and label what kind of cell we're looking at, because we're going to talk about other cells here in a minute. This is going to be a eukaryote cell. Now, eukaryotes, what do they have? They have a nucleus, so we'll have to make sure we look at that. And then I have to name my starting cell. So think about yourself. Where did you come from? You came from your parents. And so my starting cell is called my parent cell. Okay, starting cell is parent cell. So let's draw my cell. Here's my cute little cell membrane. Here is my nucleus. Let's give it some DNA. And there you go. We'll have some DNA, and then here's an organelle. Look at that. Okay, we have a parent cell, right? Now, oh no, this cell is in need. I need to make a new cell. Need to make a new cell. Okay, that's my entire job. So I have to start with my first phase. So I enter interphase, which means I get to start with G1, because G1, SG2. So what happens during G1? G stands for grow. So look, my cell literally got larger. See, this thing grew. So cell grows. I still have a nucleus. And then what else happened? Oh yeah, my organelles doubled. So organelles times 2. And then I still have my DNA inside. That's all G1 is. Say my cell grows, and then I double my organelles. Now, real quick, say this is kind of one of those like alarm situations. Hey, I just doubled something. That means I need to make sure I doubled it correctly. This is called a checkpoint. Okay, so a checkpoint is exactly like quality control. I want to make sure that I doubled my organelles correctly. And so I go to a checkpoint. If I pass the checkpoint, that means I get to move on. If I don't pass the checkpoint, that means I get sent back. Or a really crazy thing, it's called apoptosis. Let's spell it again. Apoptosis. And apoptosis is cell suicide, which just means that cell, we talked about markers being on the cell membrane. Well, a marker that I can turn on is saying, hey, I need this cell to die because he's not dividing correctly. And so these checkpoints are really, really critical, okay? All right, but let's say the cell passed. Yay, good job, cell. So now I get to go to my next stage with an S. So here's my cell. Here was my nucleus. I doubled my organelles, but now I'm going to double my DNA. So instead of having 2 yellow sticks, I'm going to have 4 sticks, and I made them in a different color so that way you can see that I replicated it. So now my DNA doubles. Look at that. We did great. Now I get to go on to my next stage, which is G2. Don't forget G stands for grow, so my cell got bigger, right? So cell grows. Look at that. Say he physically got larger. I still have 2 organelles inside, and then I still have my replicated DNA. So now this guy is going to get ready to divide. Okay, that's what he's going to do now. So now I'm going to enter M phase. Remember, M was kind of like movement. I'm going to move stuff. But another alarm, super quick. I just got through doubling my DNA. So here's my alarm. Now I have another checkpoint, which is still quality control. And again, if I check it, that means I get to move on. If I don't pass it, that means I either go back or I get destroyed through apoptosis. And so I have a G2 checkpoint. I also have a G1 checkpoint. Now let's say my cell did amazing and he did exactly what he was supposed to do. And we're going to go through M phase another day, but literally what happens is your cell starts to split. So I've made 2 nuclei. I'm going to put an organelle on either side. I have DNA in both cells. Okay, so the entire goal of M phase is to divide my DNA. And now that I've done that, I get to go through my last stage, which is cytokinesis. In cytokinesis, my cell finally splits. So I have two cells, both with a nucleus, both with an organelle inside, each with an identical copy of DNA. So that means in the end, I get two identical cells, right? These cells, because they came from a parent, these are called my daughter cells. I just made two daughters. But if you look at those cells right here versus the one I started with up here, they are identical. Okay, now, real quick, that was a eukaryote cell, right? We do have other cells on this pretty little earth. So star, 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 star. Let's talk about prokaryotes. Okay, prokaryotes, these guys can't do M phase because M phase is division of a nucleus. Prokaryotes don't have a nucleus. So they can't do M phase because they don't have a nucleus. Okay, so they go through another process. They do binary fission. Okay, and it's the same idea. Like this is the same idea. I just can't, I'm still going to divide. I just can't do M phase because of the whole nucleus situation. So if I drew a pretty picture, here's my cell, and then let's see, here's my stick of DNA. And so now my cell is going to get a little bit bigger. I'm going to double that DNA. And then my cell is going to split, and they each have DNA inside. So it's the same idea, exact same idea, but I don't have a nucleus. And since I don't have that division of labor, that means I'm going to have to, I get to do it a little bit faster. All right, now way up here, I kind of forgot to mention this, but I want to make sure I do. When I'm looking at this G1, right here, say some cells, say, whoops, let's do that. Some cells are stuck at G1, which means they're forced to go to another phase called G0. That 0 is not. So G0. These G0 cells, these never get to divide. Never get to divide, because these cells that we talked about over here with the cell cycle, with the interphase and M and cytokinesis, this would be like a body cell. Like you just made a new skin cell or a new liver cell, or you made a new egg, or you made a new sperm. But some cells never get to divide. And if you think hard, I think you would figure it out. But these would be like brain cells. You know, we can't regenerate brain cells. If you get brain damage, you have to relearn how to do stuff. You don't get to make new cells. Or if you think of somebody who has been in an accident and they become paralyzed, like spinal cord, you don't get to fix that. Say those cells are stuck in G0. And because they never divide, that means they never heal, which means you can't fix whatever the problem was. Now, one other little thing, kind of taking one more turn. So let's do some stars. Star, 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 star. Let's talk about cancer for a second. Because this stage, the cell cycle, right? This is a failure in the cell cycle. Like that's what cancer is it fails in this actual cell cycle, right? And we won't get into all the genes and stuff, but cancer by definition is uncontrolled cell growth. Okay? And so if I start doing this cell cycle and there's no checkpoints, right? There's no checkpoints involved, that means my cells are just going to keep growing and growing and growing. They're uncontrolled. This is going to result in a tumor, right? And a tumor is just abnormal growth. Like that's all it is. You do have two different kinds. We've heard these before. You have benign. Benign means it's not fast growing, and there's zero damage to anything around it. But as soon as that tumor, as soon as that growth becomes super fast growing and it starts doing damage to things around it, that's when we get to call cancer malignant. So this is fast growing and it's also causing damage. But cancer is a failure in the cell cycle. So just to recap, I have DNA. DNA has a bunch of different forms. If it's loose, it's called chromatin. If it's coiled up, it's called a chromosome. And since I've replicated, it's called a replicated chromosome. I have the specific structures right there. I have the telomeres to keep it from unwinding. I have the centromere to keep that replicated chromosome together. The cell cycle makes new cells. I have 3 stages: interphase, cytokinesis. Interphase, I want to double cell contents. I want to divide my nuclear contents. Cytokinesis, that's the final division. And then you have the pictures. Prokaryotes get to do a similar thing, except since they don't have a nucleus, they get to go through binary fission. And then a failure in all of this is going to result in cancer. All right, I hope that helps.